We have talked about motor protein that moves along actin filament, the myosin we just studied in the previous module. Now let's talk about motor proteins that move along the microtubules, kinesins and dyneins. I'm going to talk about kinesins first. Kinesins have some similarities with myosins, which suggests that they probably evolved. Kinesins and myosins, they evolved from from the same source. So kinesins was first identified in the giant squid axon, where it carries membrane enclosed organelles away from the neuronal cell body towards the axon terminal by walking towards the minus end of microtubules. Kinesin, as I mentioned, is structurally similar to myosin 2. It also has two heavy chains, two light chains, and the motor domain is again a globular domain. It has two motors and an elongated coil domain for heavy chains to dimerize. Kinesin is a member of large protein superfamily for which motor domain is the only common element. So other than motor domain, the rest of the molecule is different. Sequence of amino acids of rest of the molecule is different. Yeast has 6 distinct kinesins. Nematode has 16, and humans have 40 different types of kinesins. There are at least 10 families of kinesin-related proteins, abbreviated as KRPs, in a kinesin superfamily. Most have motor domain at the end terminus of the heavy chain and walk towards the plus end of the microtubule. As you know, there are always exceptions. We'll talk about them too in the next slide. Like myosin superfamily, only the motor domains are conserved. Conventional kinesin has motor domain at end terminus of heavy chain. Middle domain forms the coil mediating dimerization of C-terminal domains, which forms tails which attach to the cargos, such as membrane enclosed organelles. So here on your slide, you can see this is a typical kinesin with a motor domain, globular motor domain at the end terminus. Then these long coiled domains which help dimerize this molecule. And the C terminus has the domain which attaches to the cargo. C2 is a member of family of C-terminal kinesins which travel in the opposite direction from other majority of the kinesins. They travel towards the minus end instead of the plus end of the microtubule. KIF2 has a motor domain in the middle of the heavy chain. Here is your KIF2. The motor domain is in the middle of the heavy chain rather than at the amino terminus or the end terminus. Furthermore, I would like to mention about a few exceptions in kinesin family. KIF2 is a member of kinesin family that have lost their typical motor activity. Instead, they bind to microtubule ends to increase the dynamic instability. Now, let me show you about how these motor proteins, kinesins, move along their substrate, the microtubules, and we can compare them. This slide compares their activity with myosins, for example. This circle, gray circle, basically shows the amount of time myosin stays attached to actin filament. It stays attached about 5% of the time during the cycle of during its cycle moving along the actin filament. In kinesins, one head of kinesin stays attached to the microtubule 50% of the time, and the other head stays attached to the microtubule 50% of the time. So here you have the schematic which shows you this is the myosin. These are the two heads which are identical, but they're shown in green color, so you can appreciate how, or you can understand and identify also how these two motor domains switch places. In the first part, as you can see, the dark green head is attached to position number 2 on this microtubule, and this light green head, which is attached to ADP, is behind the green the dark green this head of this kinesin. The plus end of this microtubule is in this direction, so kinesin will also move towards the plus end. In the next stage, the ATP, the dark head, which was initially in the front, attaches ATP. This causes a conformational change. Now the light green head has advanced. It is ahead of the dark green head. In the next stage, this light green head attaches to the microtubule on the position number 3, if you would notice. In the subsequent change, the dark green head hydrolyzes ATP into ADP. It detaches from the microtubule, and now the light green head is ahead of the dark green head, and we have again the similar situation where we started from one head is attached, the other head is detached and bound to ATP. This process repeats itself over and over again. Consequently, the motor protein moves towards the plus end of the microtubule, tagging along whatever is attached to the C terminus or the cargo domain of this motor protein. Dianins are a family of minus end directed microtubule motors, but they are unrelated to kinesin superfamily. Dianins have 2 or 3 heavy chains, including the motor domain. Their motor domains are large, and they associate with variable number of light chains. I would also like to point out that dianins are the largest of known molecular motors. They are also among the fastest. Axonemal dianine can move microtubules in a test tube at a remarkable rate of 14 microns a second. In comparison, the fastest kinesin can move their microtubules at a rate of about 2 to 3 microns a second. So dianins are about 5 to 7% faster than kinesins. Cytoplasmic dianins are found in all eukaryotes, and they're important for vesicle trafficking as well as for localization of Golgi apparatus near the cell center. I'll show you micrographs in a different module, which clearly shows that these dianins play an important role in localization of Golgi apparatus towards the center. Axonemal dianine, which are found in cilia or flagella, they are heterodimers or heterotrimers with 2 or 3 motor domain heads. They're specialized for rapid, efficient sliding movement of microtubules that drive the beating movement of cilia and flagella. Here in this micrograph, it's a freeze etch micrograph of a cytoplasmic dianine. Here you can see this dianine has 2 heads versus this dianine, the ciliary dianine, has 3 heads. We'll continue our discussion of motor proteins in the next module.